So we have covered already that the neutrophils are the first cell types which will reach at the site of infection from the blood circulation, right? Now, we also discussed that these neutrophils can perform the process of phagocytosis. In today's class, our focus will remain over macrophages because we know the second major type of cells and the big cells which will perform this process of phagocytosis. These are called macrophages. So why I'm showing you this particular diagram, if we already have covered this, so that you can observe the visible differences between these two cell types. So these are neutrophils, right? So neutrophils are having a multi-lobed nucleus. All right, they are having two type of cytoplasmic granules or particles. I told you primary and secondary granules, right? And there is, if you see, there is no or almost very small few Golgi apparatus and rough endoplasmic reticulum in neutrophils. So they cannot divide actually. They cannot form the proteins which are required, or they can synthesize very less number of proteins because of their limited Golgi body and rough endoplasmic reticulum, where the new protein synthesis or translation is actually taking place. Now we will discuss about macrophages. So let's see what is the structure of a macrophage. So in blood or in the blood circulation, the cells which are present are called monocytes. These monocytes will go into the tissue spaces or at the site of infection by the same process by which neutrophils will move. So this is called extravasation and then chemotaxis. Finally, these monocytes will reach at the site of infection, where they will differentiate into large macrophages. And these macrophages are given separate names. They are called according to the tissue in which they are present. For example, in the brain, in our CNS, they are called microglial cells. In our kidney, they are called mesangial cells. In our liver, they are called Kupffer cells. So each time when you talk about a tissue, these macrophages will have a different name. They are specialized cells and they named according to the tissue in which they are present. But overall, they are having the same function because they are macrophages, right? So they can perform the two major functions, but at least we discuss some other functions. For example, these macrophages are important in tissue repair and remodeling. Whenever we are having an injury or wound, so these macrophages will help in removing the pathogens or dead cells. Not only this, they also help in tissue healing and repair of tissue damage. So actually they are having multiple functions, but from the viewpoint of immunology or immune responses, their main function is phagocytosis because of their large size, they can easily engulf the bacteria, as you can see in this particular diagram. So this is a macrophage which is taking a group of bacterial cells or a colony or a bunch of cells, right? So they can uptake many bacteria simultaneously because of their large size. Generally their size range, so you may ask me a question, what is actual size of these macrophages, so that they can uptake not a single but a group of bacteria. So generally they are having size range 20 micron, 20 to 30 micron, depending upon situation their size can even up to 30 microns. Generally eukaryotic cell is having size range 10 to 20 micrometer, and a bacterial cell is prokaryotic cell is generally having size less than 1 micrometer, right? So now you can assume that what is the size difference between a bacterial cell or a pathogen and the cells which are responsible for removal of these pathogens or foreign particles. So generally they are having size range 20 to 30 micrometer, micron or micrometer, whatever you call it. All right. So okay, so why I'm showing you this particular diagram. So please note this. Actually, they are having a big nucleus. Unlike neutrophils, they are having a single nucleus. There is no lobe in the nucleus, and their nucleus is covered with this nuclear membrane and this rough endoplasmic reticulum. They are having uh, rough endoplasmic reticulum where ribosomes are attached and the protein synthesis is actively taking place. Generally, a macrophage can synthesize more than 100 different proteins, which are important in immune responses, right? Second thing is they are having Golgi body. So obviously Golgi bodies are required, or Golgi apparatus is required in protein um, maturation or processing. Generally, these glyco means the carbohydrate components are called glyco. They are attached over proteins. The proteins are synthesized over these rough endoplasmic reticulum, and the protein will move to the Golgi body where they are glycosylated and processed so that they can be targeted to either lysosomes or to the membrane, or in severe cases they can be secreted out of the cell, right? So they are called secretory proteins. So because they are having Golgi body and rough endoplasmic reticulum, they can synthesize a large number of proteins. We will discuss some very important proteins which are secreted by these macrophages. So first of all, you are able to understand the structural difference between a neutrophil and a macrophage. So neutrophils are generally small in size and they do not have these rough endoplasmic reticulum and Golgi body in a large number. But the macrophages, they are not only big in size, they also having the actively translating regions in the cytoplasm so that they can synthesize large number of proteins which are actively taking part in the immune responses. Therefore, neutrophils are small phagocytic cells, macrophages are big phagocytic cells. So generally neutrophils are called chote havaldar and macrophages are called bade havaldar because they can kill the large pathogens. Not only this, they are also having one very important property we will discuss. So at time, if pathogen is big enough, which cannot be engulfed by a single macrophage, so what will happen? Several macrophages will fuse together to make a giant cell, and this big cell then will kill the pathogen anyhow. So they are also having this fusing property to make a big cell. So we will discuss that thing also. So first of all, we have covered this already. So depending upon the tissue in which the macrophages are present, macrophage cells are present, they are having a specific name. For example, in brain, they are called astrocytes or microglial cells. They are in the lungs, they are called dust cells. In the skin, they are Langerhans cells. In the epidermis, okay, in the liver, they are Kupffer cells. In the spleen, spleen macrophages. So these spleen macrophages, you know, RBC's graveyard is the spleen, in which the RBC will move after 120 days and they finally will be degraded. So these spleen macrophages are actually responsible for removing or killing the red blood cells which are present in our blood after 120 days, right? Then there is intestine where we are having M cells or microfold cells. They are specialized macrophages in intestine. In lymph nodes, we are having follicular dendritic cells, FDCs, right? The placenta, we are having Hofbauer cells. Connective tissue, there are histiocytes. In the blood, there are monocytes, but in the tissues, they are generally called macrophages. In the kidney, there is mesangial cell. In the bone, there is osteoclast. All these things we already have covered. Okay, so in the heart, myocardial cell, for example. So I was telling you that macrophages may live for a long time after ingesting the chemically inert particle. Suppose some particle, this macrophage or bade havildar have ingested some particle which is not ingested, which will not be degraded. So in such cases, what will happen? The lifespan of macrophage will increase. So by ingesting the chemically inert particles, such as the carbon injected into the tattoo marks. Okay, so generally the ink is used in the tattoo, and these ink particles or the carbon atoms are uptaken by the macrophages, and because they are chemically inert, so it can take time to degrade them. Ultimately, what will happen? The lifespan of macrophages will increase. They may live for a long time, right? Or in several cases, they may fuse to form a giant cell, and in an attempt to eliminate those foreign material. All right. In some circumstances, such as in smokers, macrophages may carry the particles to the lungs or to the intestine, and from there into the bronchi or into the intestinal lumen, from where they can be eliminated. They are eliminated, they are removed. Some foreign particles are toxic to the macrophages. For example, asbestos, right? So in such circumstances, if macrophages uptake some toxic substance, what will happen? It will kill the macrophage. Once it will perform the phagocytosis, right? And when this macrophage will die, it will release this asbestos, because it will not be degraded by the macrophage, and it will kill the macrophage. Macrophage then, after it will release out of this dead cell, the particles must be repeatedly rephagocytosized. So other macrophages, nearby macrophages, will uptake this asbestos, and each time they are toxic to the macrophages. It will kill the macrophages, leading to the release of lysosomal enzymes. So in such circumstances, what will happen? Because of the death of these macrophages, large number of lysosomal enzymes, which are called suicidal bags of the cell, will be released in the tissues, and reactive oxygen metabolites will be released, because nitrogen and oxygen intermediate, we have discussed about reactive oxygen species and reactive nitrogen species. If you do not remember about reactive nitrogen species, so I also covered this in this particular slide. All right. So generally, all the reactive nitrogen species are formed from arginine amino acid. So arginine amino acid is actually having this nitrogen group, or this amino group, which is actually called guanidino group, right? So this will be converted into nitric oxide, which is toxic. So this nitric oxide then will fuse with oxygen to make nitrogen dioxide. Okay, and this can form N2O4, N2O3, and in presence of water, it can further form NO2 negative, NO3 negative, these anions. In some cases, this nitric oxide will fuse with the superoxide ion, that is O2 negative charge, and it will form ONOO negative, that is called peroxynitrite, right? So it can also form. What will happen? This nitric oxide will bind with some reactive oxygen species, which is also present inside the cell, called superoxide anion. So this is called O2 negative, right? This is superoxide anion. It is also having a free electron, can have a free electron to make ONOO negative. So this can result, and this is very toxic to any pathogen. It will kill the pathogen or the foreign cell. All right. So this is called peroxynitrite anion. So this peroxynitrite will be highly toxic, and in some circumstances, in presence of some H positive ions, it can further degrade into, in presence of H positive ions, it will be degraded into hydroxy radicals, that is OH negative, or a free electron over the oxygen. So hydroxy radicals, which are also toxic to the foreign pathogens, they can result. So you can see that reactive oxygen species and nitrogen species intermediates, they are connected pathways, and they can give rise to highly toxic intermediates. For example, nitric oxide, NO2, NO2 negative, NO3 negative, peroxynitrite, or hydroxy free radical. And in fact, this oxygen species, that is reactive oxygen species, this is also very, very toxic. So what will happen when these phagocytes, then when these macrophages will kill by asbestos, they will release these nitrogen and oxygen species into the tissues, and it will harm the tissues. And such condition is known as asbestosis, in which uh, the tissue destruction will take place. So this condition, this disease is known as asbestosis, in which the patient will uptake either asbestos particles or all right. Okay, because this asbestos is toxic to the macrophages, they will kill the macrophage, and each time when the macrophage is killed, their contents, their reactive oxygen species, nitrogen species, and their enzymes, lysosomal enzyme will be released into the tissues, leading to a condition which is harmful to the tissues, in which tissues will be destructed, is known as asbestosis. All right. Now, so what is the lifespan of a macrophage? Macrophages are also produced from the same stem cells in the bone marrow from, uh, so I will show you one more diagram before going into the details about the lifespan, how these monocytes and macrophages are produced within the page number 28 of your QA. There is a, we have discussed this pathway many times, and this is very, very important in fact, because during hematopoiesis, which is a self-renewing cell, the stem cell will give rise to two types of cells, myeloid progenitor and lymphoid progenitor. Out of lymphoid progenitor, the lymphoid cells, BT and NK cells will be produced along with dendritic cells. And in case of myeloid progenitor cell, it will give rise to different types of cells in the blood. For example, granulocytes, neutrophils, eosinophils, basophils. Basophil can also give rise to mast cells, and it can also give rise to the same cell. Myeloid progenitor will give rise to the megakaryocytes. These megakaryocytes will give rise to the platelets, which are not complete cells but cell fragments, and red blood cells. Out of these are called erythrocytes. Not only this, they can also give rise to some other cells which are called monocytes. So see this, this granulocyte monocyte progenitor will either give rise to neutrophil or a monocyte. If it is monocyte in the blood, when it will move to the tissues, it will differentiate into the macrophage, or it can also differentiate into a dendritic cell by this. Okay, so this monocyte is actually coming from this stem cell. So now see this particular diagram, the second one, which I already showed you. So this stem cell, the stem cell from where the monocytes or the macrophages are coming, these stem cells are called monoblast. In the bone marrow, monoblast will be giving rise to promonocytes, which are having a life of 6 days, right? Then these promonocyte will from the bone marrow will come into the tissue circulation or in the blood circulation or in the blood. In blood, they are called monocytes. So promonocyte will be converted into the monocyte. Monocytes are generally having 1 to 2 days lifespan in the blood, and then they will move to the tissues. In tissue, as I told you, they are having different names, but all of these are macrophages. So they're called osteoblast in the bone or osteoclast. All right, macrophage in the tissues, they are called Kupffer cells in the liver, mesangial cells in the kidney, microglia in the brain, histiocytes in the connective tissue, right? So in each tissue, they are having different names, but all of them are macrophages. And in tissues, they can survive for months, right? All right. So the major function of these cells now is to perform the process called phagocytosis. So sorry. Actually, we are here. So one of the major function of these phagocytes or these macrophages is to perform the phagocytosis. So first we will discuss about all right. So the myeloid progenitor cell, as I showed you in the diagram, give rise to a group of phagocytic cells called monocytes, which will be differentiated into the macrophages and dendritic cells. These are professional antigen presenting cells functions, right? So they are having this function. By this function, they can activate other cells of the immune system. For example, lymphocytes, B cells, T cells, right? They can be activated by these antigen presentations. All the monocytes which are present in the blood, they make up about 5 to 10% of the white blood cells. So out of total leukocytes, there are 5 to 10% monocytes. And these monocytes are heterogeneous group of cells that migrate into the tissues and differentiate into the diverse array of tissue resident phagocytic cells. These are called macrophages and dendritic cells. During hematopoiesis in the bone marrow, granulocyte monocyte progenitor cell differentiate into the promonocyte, which leave the bone marrow and enter into the blood, where they have further differentiated into the mature monocytes. Two broad categories of monocytes have been recently identified. So there are two major type of monocytes. One is called inflammatory monocytes and second is called patrolling monocytes. So first of all, we have inflammatory monocytes and second type we have patrolling monocyte, right? So inflammatory because they enter into the tissues quickly in a response to the infection. If there is an infection, they will quickly move into the tissues. Therefore, they are called inflammatory monocyte because they will take part in the inflammatory response. Second is patrolling. So as the name suggests, they are having patrolling function. They will check, they will keep an eye over these infections. So smaller group of cells that crawl slowly along the blood vessels provide a reservoir for tissue resident monocytes in the absence of an infection. So generally they are also present whenever there is no infection. So it is just like police. So police not only take part whenever there is a crime, they also having this patrolling function. They keep an eye to control the crime, right? So the patrolling monocytes, they are a smaller group of cells, but they crawl slowly along the blood vessels, provide a reservoir for the tissue resident monocytes in the absence of infection. If there is no infection, still they are present and may quell rather than initiate an immune response. So generally, they do not initiate an immune response. They are having this patrolling function, whereas inflammatory monocyte will mount an immune response. All right. So there is one more important line. Activated inflammatory macrophages are more effective than the resting ones. Obviously, if you are resting, if you are not active, uh, you will learn few things. So once the macrophages are activated, they are actively taking part in the phagocytosis. So already we have covered the second process called opsonization, in which the phagocytosis can increase up to 4,000 fold in presence of specific antibodies to the antigen than in its absence. So these are called opsonins, either antibodies or some complement proteins, for example, C3b, C4b. We already have covered in our previous classes. They are major opsonin proteins, and they will perform the opsonization, or they will coat that particular pathogen or antigen, and they will increase the rate of, they will enhance the rate of phagocytosis by about 4,000 fold. So this is a huge increase, right? All right. So now, this is quite interesting, that why we have discussed neutrophils first and then we came to the macrophages, because neutrophils are having one more function. Neutrophils are neutrophils are important because neutrophils are the first cell types in the blood which will reach at the site of infection. All right. Then they will activate immune system, not directly, but once the neutrophils are, so let's read this particular theory so that you can understand what is written here. So phagocytosis by macrophages is similar to the process which is actually taking place in the neutrophils. All right. So this is quite understandable that the phagocytosis is the common process. Neutrophils also can do this. Macrophages can also perform this. But the difference is neutrophils are being small size and they generally do not activate an immune response. They do not present antigens to any other cells. But macrophages, they are having the role, this particular function of activation or antigen presentation. Therefore, they can also activate other cell types which are present in the immune system. Macrophages are attached not only to the bacterial and these products such as N-formyl methionine peptides and products of the immune reaction, for example, C5a. As I told you in the previous classes, generally the phagocytic cells are moving by crawling movements. So they reach at the site of infection by chemotaxis. So what kind of factors are going to attract these phagocytic cells? They are generally the N-formyl methionine peptide, because the proteins which are present in bacteria are N-formyl methionated. So methionine, this starting amino acid methionine, is actually having this formyl N-formyl formylated, so that macrophage or other cell can recognize that particular peptide, that oh, it's belong to some pathogen, because it is generally the prokaryotes having this kind of methionine. Right. Second is C5a. C5a is complement protein 5 fragment, small fragment A. So this will work as chemotactic factor. Not only this, IL-8 also will work. So in the previous class, I have covered C5a and IL-8. Both are very important chemotactic factors. So C5a, also we have already discussed about interleukin 8, which will also act as a chemo attractant or chemotactic factor, so that the cells of the immune response will move towards the site of infection or injury. Okay, so interleukin 8 and C5a both are major chemokines. Also, these macrophages, so this was, we also have discussed in case of neutrophils. Neutrophils are also attracted by C5a, IL-8 and all these things. For example, N-formyl methionine peptides, which are actually belonging to the pathogens or bacteria. But now see the difference. The macrophages, because they are major type of cells which are involved in immune responses, major phagocytes, why we are calling them major phagocytic cells, because of their big size. Not only this, they will be activated by some other means. For example, the factors which are released by the damaged cells and extracellular matrix. So this is quite important, because if some cells are damaged, suppose neutrophils will die after digesting some foreign particle. So the damaged cells or the extracellular matrix components, which include the fragments of collagen which is present in the connective tissue, elastin, fibrinogen. So if these proteins which are generally present in the tissues, or if their fragments are present, So they can also activate these macrophages, these big cells, so that they will become activated macrophages, right? And their performance will increase once they are activated. Neutrophils on dying release elastase and collagenase. So whenever there is death of neutrophil, which are small phagocytic cells, their components, including the collagen, these elastase and collagenase, they will be produced and ultimately they will be recognized. So to generate monocyte chemotactic factor. So these are called MCF, monocyte chemotactic factor. They will attract these monocytes from the blood. Monocyte will reach at the site of death of neutrophil, and they will be activated. They will be differentiated into the macrophages first, and then they will mount an immune response. They will present the antigens, they will perform the phagocytosis and so on. So neutrophils are thus a kind of martyrs in the immune response, immune system, and in our body. They reach and attack foreign material first, and in the dying, whenever they will die, they will attract the macrophages to the site of microbial invasion wherever there is an injury or infection, yeah, or inflammation. All right. So the site of invasion where the pathogen has invaded our body or the host body. Macrophages destroy the antigen by both oxidative and non-oxidative mechanism. If you do not remember, in your Pathfinder, we have already covered both processes. Okay, so I'm showing you first. So if you have this book, we already have covered both the types called oxygen dependent killing mechanism and oxygen independent killing mechanism. Oxygen dependent means there is reactive oxygen species, reactive nitrogen species. So everything we have covered. And if there is lysozyme defensin mediated damage to the host, damage to the pathogen, this is called oxygen independent killing mechanism. All right. So there is, both are functional in macrophages also. Oh no, actually I have. All right. अभी देखो यहाँ पे दो तरह. Okay, so first we will discuss resting macrophages and then we will discuss activated macrophages, right? So generally, if there is no infection, no inflammation, so these are called resting macrophages or monocytes. Generally, you can call them monocyte because these are not differentiated, right? If there is any inflammatory stimulus which is coming, some cell fragments, some kind of, for example, C5a, interleukin 8, all these are kind of inflammatory stimulus. So it will activate these monocytes or macrophages so that inflammatory macrophages, they will be converted into inflammatory macrophages. How you will characterize them? So there is an increase in the lysosomal enzyme, there is increase in the phagocytosis, expression of membrane receptor will increase. We will discuss them, how many different types of membrane receptors they are found in the macrophages, over macrophages. We'll cover this one. And neutral protease secretion. So they will secrete some of the proteins. Proteases are enzymes which are responsible for protein degradation, right? So it has been seen that protease secretion is also increased. Then, so first of all, the resting macrophage or monocyte are converted into inflammatory macrophage. Finally, this inflammatory macrophage will be converted into activated macrophage. Now, actually, the activated macrophages are the major phagocytes which will show large size. So I will tell you what are the major things. So their size will increase. They will show the increased movement towards the site of infection. Their membrane activity is increasing. Lysosomal activity will increasing. Phagocytosis is even more pronounced. Bactericidal activity, they will kill the, cidal means killing mechanism. So they will kill the bacterial cells, and they will show, so this is, last one is more important. MSC class 2 expression will increase over their membrane, and because they will express the class 2 molecules, with the help of this class of major histocompatibility complex, they will present antigen fragments to other cell types. Mainly with the help of class 2 molecules, they will present this antigen, right? So they will, sorry, I want to annotate this, but sometimes it will. All right, so what will happen? MSC class 2 molecules will generally activate which type of cells? T helper cells. So from here you will keep this into mind. So the T helper cells will interact with the MSC class 2 molecules, and if it is written like MSC class 1 molecule, so they will interact with, so this question has can be asked, already have been asked. So that's why I'm discussing this here. So MSC class 1 molecules will activate T cytotoxic cells, which are second type of T cells, major T cells. So this, by the process of antigen presentation, they will present antigen with the help of class 2 molecules. They will present to the T helper cells. Generally, macrophages do this, but some other cells, they can also present this antigen with the help of MSC class 1 molecule to the T cytotoxic cells. Now, sometimes the question is more tricky even. So these T helper cells are called CD4 positive cells, right? CD4 positive cells, whereas TC cells are generally called CD8 positive cells. So CD8 positive cells. So now you will remember them. So MSC class 1 T cytotoxic cells, which are actually called CD8 positive cells. T helper cells, they are CD4 positive cells, right? If you are not able to remember them, just do one thing. So these are H actually. So if you do this, so this is H. I'm using some other color so that you can understand what I'm doing. So what will you do? You will remove this particular fragment from here. You will add it here, right? And you will join this, right? So this is actually the CD4 positive cells. T helper cells are CD4 positive cells. In case of TC cells, generally, you will, what will you do? You will join like this. So they will become CD8 positive cells, right? So this is the simple trick how you will remember them. There is no need to ratify this. Okay. So T helper cells are actually called CD4 positive cells. TC cells are called CD8 positive cells. All right. So what we have discussed, first thing is resting macrophages, which are also called monocytes, which are present in the blood. If there is an inflammatory stimulus, they will come out of the blood and they will move into the tissues where they're called inflammatory macrophages. Or we already have discussed that there are two types of monocytes. One is called inflammatory monocyte, one is called patrolling monocytes. So these inflammatory macrophages are a result of these inflammatory monocytes. So they will come into the tissues and they will show increase in the lysosomal enzyme phagocytosis, expression of membrane receptor, and secretion of the proteins, right? If there is interferons beta, these bacterial degradation products and other stimulus will also meet. Ultimately, they will differentiate it into highly activated macrophages, and they will show increase in size, movement, membrane activity, lysosomal enzyme, phagocytosis, and they will show the bactericidal activity by secreting some of the proteins. Also, they will present antigens to some of the cells called T helper cells, so that other cells of the immune system can also be activated. Now, in this next diagram, we will discuss what major type of receptors are present over macrophages. Before that, there is a query actually. All the nucleated cells, जिन भी cells में nucleus होता है, they can present antigen with the help of this class 1. Okay. So generally antigen presenting cells, they are all nucleated, right? So they actually have both type of MSC over their surface, MSC1, MSC2. But remember, MSC1 will always interact and activate the T helper cell, sorry, T cytotoxic cells, because MSC1 will bind to CD8, and CD8 is actually present over T cytotoxic cells, TC cells. But MSC class 2 molecule will interact with CD4, and CD4, which is a cluster of differentiation for, which is present only over the surface of T helper cells. Right. So all the nucleated cells are actually having this class 1 molecule, MSC. So biologically, antigen presenting cells, which are actually macrophages, B cells, and dendritic cells. So these are called professional antigen presenting cells. All three are nucleated cells, so they actually also have MSC class 1 molecules over the surface. Right. If you also have some query, please ask. I'm giving you a 2-minute break so that we can start. What kind of receptors are generally present over the surface of macrophages? All right. So we will discuss this. If you have any query from this much we have discussed, please ask. Because all are receptor for immunoglobulin G class, therefore they are called FC receptor for gamma. This is receptor 1, receptor 2, receptor 3, according to their affinity to the antibodies. So generally receptor 1, they are called CD64 because they are having high affinity for the antibodies. And if you remember, in neutrophils, we are only having CD32, not CD64. So generally, they are having less affinity for antibodies in comparison to macrophages, because macrophages can bind with antibody with very, very high affinity because they are having CD64, which is actually called major receptor type, receptor 1 for immunoglobulin G antibodies. Second thing is there are other type of receptors also. So this is one thing. Because they are having high affinity receptors, macrophages can bind antibodies with high affinity in comparison to neutrophils. Second thing is they are having complement receptors. So also neutrophils also have. So complement receptors, they are going to bind with the complement fragment C3b or C4b. Generally, they bind with C3b, right? Also, they are having the CD16B and CD18. It will bind with the fragments of C3 fragment, that is called D, C3D and C3DG. This is small actually, sorry for this, C3DG, right? So these are fragments, even smaller fragments of C3B. They are called C3D and C3DG. They will bind with this particular CD11B, CD18 heterodimer, right? As you can see, complement will bind to this one. They're also having receptor for interleukin 2. As I told you that after antigen presentation, they will activate the T helper cells. These T helper cells will secrete interleukin 2, which will bind to other macrophages which are still not activated, and it can activate other macrophages also. So this is called CD25, which will bind with the interleukin 2 secreted by the cells of immune system, so that it can activate more number of macrophages. Also, they have CD71 for transferrin. They are having this B7 receptor. This is very important. I will discuss this B7 too, which will actually bind with CD28 whenever we are having a major class over antigen presentation. I will tell you how these B7 and CD28 interaction is important, right? These B7 and CD28 interaction are also important whenever these antigen presentation is actually taking place. So generally, CD4 molecules which are present over T helper cells, they will bind with the MSC class 2 molecules, which will be expressed over. So let me draw something so that you can understand this better. So they will express MSC class 2 molecules over the surface of macrophage. So this is a general monocyte or macrophage which is having all these receptors. So this is a macrophage cell, and it will represent the antigen. So it will express some kind of receptors over its surface, and these receptors will have some kind of antigen, right? So suppose this is having a fragment of antigen. This is the fragment of antigen, which it is expressing to some other cell. So that because this is MSC class 2, obviously it will bind with, it will interact with CD4. All right. So T helper cell will come and it will recognize this MSC class 2 molecule with the help of its CD4. So this CD4 is actually, which is present over this T helper cell, will bind with this MSC class 2 molecule, and it will activate this T helper cell. Also, this T helper cell will have the CD28, which will recognize the second receptor over its surface, that is called B20, B7. Right. So this B7 CD28 interaction is also very, very important whenever there is an interaction between antigen presenting cell and the other cells of immune system, so that it will generate two type of signals, one from here and one from here, so that it will activate this T helper cell, and it will secrete some other cytokines, for example, interleukin 2, interleukin 4, all these, to activate either other macrophages or other cells of immune system, right? Interleukins, a general term. For example, interleukin 2, interleukin 4, all these are secreted from the T helper cell, and they will, for example, interleukin 2 receptors are present over macrophage itself or monocytes. They will activate the monocytes into highly activated macrophages. So that's how this pathway, this mechanism is performed by the immune cells and immune system. All right. Got it? So that we can move to the next diagram. Now, if you look into the functions or the roles of macrophages, they are having diverse functions. A single macrophage can take part in the tissue breakdown. They will remove all the cells from the damaged tissue. They will secrete collagenase, elastase, plasminogen activator, fibroblasts. They will activate the fibroblasts, so that fibroblasts will also secrete collagenase enzyme. So this collagenase will actually digest the collagen in the connective tissue. So this is one arm of this. That's how they are able to remove the dead cells and foreign particles, right, with the help of all these enzymes. The second branch is, the second arm is showing they're also beneficial in the tissue remodeling. They are actively taking part in tissue remodeling processes. So they are important in tissue remodeling to make new cells. Uh, Fibroblast growth factor, they will secrete some of the fibroblast growth factors and increase the collagen synthesis. Angiogenesis factors. So what is this angiogenesis? Angiogenesis is formation of new blood vessels. So macrophages will secrete some molecules which are regulating the angiogenesis. So these angiogenesis factors will neovascularization, responsible for neovascular. Neo means new, vascularization means new blood vessel formation, so that new tissue growth will take place. So this is called tissue remodeling. So they are also actively taking part in tissue remodeling. Okay, so control of synthesis in the connective tissue matrix proteins. Some of the matrix proteins in the tissues are also under the control or regulation of macrophages. They actively take part in tissue remodeling and removing the dead cells from the tissues. All right. So next diagram is showing, oh, this already we have covered actually. So indeed, almost 100 different proteins are secreted from these macrophages, and they are taking part in antigen presentation, phagocytosis, destruction of bacteria or pathogen. They will remove the tumor cells. Okay, so, or the dead cells from the tissues. They're also responsible for tissue remodeling and reorganization. very important in our wound healing. If we have an injury, macrophages are actively taking part in tissue remodeling and healing of our wounds. They secrete a large number of proteins. Around 100 proteins are secreted, so including some cytokines, enzymes, and other factors. Cytokines, some very important cytokines which are secreted are interleukin 1, 6, 12, tumor necrosis factor alpha, interleukin 8. As I told you, interleukin 8 is a kind of chemotactic factor, and granulocyte monocyte colony stimulating factor. This we already have covered in our, so if you remember this particular diagram, oh, where it was, 28, I think. Okay. So the granulocyte monocyte colony stimulating factor is responsible for the differentiation of this myeloid progenitor into the granulocyte monocyte progenitor, so that it can be differentiated either into the monocyte or into neutrophil, eosinophil, basophil, right? This GM-CSF is actually responsible for the synthesis of granulocyte and monocytes. In granulocytes are three major type of cells: neutrophils, eosinophils, and basophils. Monocytes will develop into them either macrophages or dendritic cells. Some enzymes are also actively secreted out of these macrophages, including lysozyme, acid hydrolases, arginase, lipase, collagenase, elastase. They will either kill the pathogen or they will remove the dead cells or these cancer cells from the tissues. Other factors, for example, prostaglandins, leukotrienes, complement components, fibronectin, binding protein, coagulation factors, these are also secreted out of the macrophages. So out of these, prostaglandins and leukotrienes are major type of chemical signals due to which we are able to feel the pain. So our pain sensation is due to the secretion of these prostaglandins and leukotrienes. Okay, so whenever we feel ill, we are having some fever-like symptoms, it is due to the increased production of these prostaglandins and leukotrienes. So majorly prostaglandins are responsible for pain sensation. So obviously the macrophages are activated in response to some infection or injury or in the inflammatory responses. We do have an increased synthesis of prostaglandins and leukotrienes. They are secreted actively out of this macrophage. So they will develop the symptoms of illness and we will feel the pain and fever, right? Now, as we already have discussed that the cytokines which will be secreted by these macrophages are interleukin 1, 6, and 12, along with tumor necrosis factor alpha. So these are very, very important cytokines. They will not only having single function, but they are having diverse functions, right? So first of all, we will discuss interleukin 1. Interleukin 1, which is actually secreted from macrophage, it can activate the T cells, B cells, and natural killer cells. It can activate all different types of lymphocytes. So T cells, once they are activated, they will there is increase in IL-2 production, IL-2 receptor expression, interferon gamma production, and interleukin 3 production. So interleukin 2, interleukin 3, and interferon gamma production, which is from the T cells in response to interleukin 1. Now you will note this. Actually, the T cells are having both kinds of receptors simultaneously. They will secrete interleukin 2, they will produce interleukin 2, and they're having a receptor for this interleukin 2 along with macrophages. So macrophages also do have this interleukin 2 receptor. The T cells are also having this receptor, whereas the B cells are actually having interleukin 4 receptors. So they will synergize with interleukin 4 and 6. Also, there is an enhances in the proliferation, cytotoxic activity, and chemotaxis. Chemotaxis is the movement of blood cells towards the site of injury or inflammation. If the B cells are activated in the response of interleukin 1, they are showing increased growth factors, interleukin 4, interleukin 6, because these B cells are having interleukin 4 receptors. So the T cell also secrete the interleukin 4 actually. So they will bind with the B cells and they will activate the B cells. Enhancement of antibody production. B cell will, after activation, they will be transformed into the plasma cells. The plasma cells will show antibody production. Chemotaxis and maturation, right? Natural killer cells, they are having same role like interleukin 2 and interferon gamma. So they will be activated with the T cell will secrete these molecules and they will activate the natural killer cells. They stimulate the tumor lysis. They will kill the tumor cells. They will cytokine synthesis. So different types of cytokines are secreted out of these natural killer cells. All right. Then the second one is interleukin 6. So the one we already have covered. 6. Actually, the interleukin 6 is secreted by not only by macrophages, it can also be secreted by different types of cells. So here is macrophages. So macrophages are one type of cell which can secrete interleukin 6. But interleukin 6, because it is very important cytokine, can be secreted from the B cells, T cells, fibroblasts, keratinocyte, endothelial cells, or even bone marrow stromal cells. There are some specialized cells in the bone marrow called stromal cells. They can also secrete this interleukin 6. Now, what is important, because we know the macrophages are also secreting interleukin 6. Now, what is the role of interleukin 6? It is very diverse. It will activate the B cell, hepatocytes, neurons, macrophages. It will allow them to differentiate. T cells, differentiation, activation, interleukin 2 production. They will activate the bone marrow stem cells so that they can proliferate and differentiate, right? So there are large number of functions, right? In the hepatocytes in the liver, they will stimulate the activation, the production of acute phase proteins. So acute phase proteins are kind of proteins which are synthesized by liver, and they will come into the blood circulation and they will require the time of injury and inflammation, right? In case of B cell, they will allow them to differentiate and immunoglobulin synthesis. So B cell will differentiate it into the plasma cells. And as I showed you, as I told you, the B cell will, once they will mature into the plasma cell, they will secrete large number of antibodies or immunoglobulins. Then these macrophages also secrete interleukin 12, which is very, very important. Not only macrophage, B cell and dendritic cells, which are, okay, so all these three cells are professional antigen presenting cells. As I told you, professional antigen presenting cell. So we can put it here so that you can remember professional antigen presenting cells, or simply APCs. So all these three, macrophages, B cells, and dendritic cells, all, what is the common in all the three cells? They will secrete the interleukin 12, which is a kind of cytokine. Now let's look into the functions of this interleukin 12. So it can activate the T cells. It can also activate the natural killer cells, which are major two types of lymphocytes, right? And T cells, they enhance the cytotoxicity, their proliferation, their interferon gamma production, and they will suppress the IgE formation, whereas they will increase the other kinds of. All right, so this IgE is actually produced by the B cells, right? Once the interleukin 12 is produced from the B cell, it will suppress the immunoglobulin E production, E formation, whereas other type of antibodies will be generated. So we already have covered there are four type of antibodies which are produced by B cells, but IgE formation will be suppressed in case of interleukin 12 production. Natural killer cells, they enhance the cytotoxicity and interferon gamma production. In cytotoxicity, they are responsible for killing the tumor cells. They call natural killer cells. Then there is tumor necrosis factor alpha, TNF alpha, which is produced by not only macrophages, but also by the natural killer cells and T cells. So this tumor necrosis factor alpha is responsible for killing the tumor cells. As we know, the T cells, T cytotoxic cells, are responsible for removing the cytotoxic cells or the tumor cells. The TC cells are responsible. So these will secrete the tumor necrosis factor alpha. Tumor necrosis factor alpha can have various functions. For example, they will, first major one is actually to destruction of tumor cells. So first of all, I'm covering this. The tumor cells will be removed from the body with the help of this tumor necrosis factor alpha, which is also called tumor necrosis factor TNF. Sorry, it is hard to write using the. All right, so you will call it TNF alpha, right? It will have major function of removing or destructing the tumor cells which are present in the body, which can show unregulated proliferation. This tumor necrosis factor alpha will activate the muscle cell and increase their catabolism. Adipocyte also affected by this, but this is not a very important. They will activate the neutrophils. This is important. They will increase the phagocytosis, right? The tumor necrosis factor alpha will activate the neutrophils and they will increase the phagocytosis. They will activate B cells, allow them to differentiate and growth. They will allow the fibroblasts to proliferate. They will allow the endothelial cells, which are making the blood vessels or blood walls, to produce interleukin 1. And as I showed you in the previous diagram, then this interleukin 1 will have various kinds of effect, right? They will increase the catabolism in hepatocytes or liver cells in the osteoclast. They will allow them to activate. These are macrophages which are present in the bones. They will allow them their activation. So once this interleukin 1 is produced again from the endothelial cell in response to the tumor necrosis factor alpha, what can happen? Again, the same, everything can happen whatever we have covered in this particular diagram. So after this interleukin 1 will be produced by the endothelial cells, it can activate the B cells, T cells, and natural killer cells, right? And they will show different. So by now you are able to understand that a single cell is not working alone. It is working in coordination with other cells, and it is actually activating other cells or suppressing other cell types. So up to here we have discussed about three major types of, or four major type of cytokines which are belonging to macrophages. These include interleukin 1, interleukin 6, and interleukin 12 along with tumor necrosis factor alpha, right? So जितने भी cytokine आज हमने discuss करे हैं, these all are immune activating cytokines. They will activate the immune response. So immune response will be increased in their response, in their production. Immune response will increase. If someone asks you what cytokine is mainly responsible for decreasing or regulating the immune response, which is already high, so the answer is interleukin 10. So it is actually responsible. It is having an opposite effect. So all the cytokines which we have discussed and interleukin 10, they are having opposite effects. They will these whatever we have discussed today, they will increase the immune response, but interleukin 10 will try to suppress the immune response, right? So this arrow will become down in response to interleukin 10. So each, what the points are written here, we already have covered. Macrophages phagocytosize particle in the similar manner, which is actually neutrophils do, but they do so by repeatedly and live much longer. So actually neutrophils can phagocytosize only once and then they will die. And their fragments, whatever the fragments are produced out of neutrophils, dead neutrophils, they will activate the macrophages. Macrophages are different because they are having large size. They can perform the phagocytosis event repeatedly, and they will live much longer than neutrophils do. Actually, they are present in the tissues, and in tissues, they can survive 4 months. Right? Whereas neutrophils are having lifespan generally lasts for about 1 day or 2, or they are having lifespan in hours. Macrophages are secretory cells. This is also very important because they're producing large number of proteins. Around 100 proteins are produced because they are having Golgi body and rough endoplasmic reticulum. They produce protein that influence the inflammation. They are taking part in healing, wound healing or tissue repair, and the body's response to the infection. They will activate other cell types. So they will regulate this body's response to an infection or an injury, right? Most important of these are cytokines, interleukin 1, 6, 12, and tumor necrosis factor alpha we already have covered. Macrophage-derived cytokines can cause a fever and are responsible for many of the clinical signs of infection. As I told you that these are responsible for producing the prostaglandins and leukotrienes. They are having this pain and fever symptoms in the host body. Macrophages in the spleen, liver, and bone marrow are largely responsible for removing the foreign particles. So macrophages which are actually present in the spleen, they can kill their RBCs also, but they are responsible for removing the foreign particles or pathogens from the blood, because during the circulation, blood will move, blood will go to these organs, such as bacteria from the bloodstream. So they will remove the pathogen from the blood in the macrophages which are actually present in the liver, spleen, and bone marrow. In the liver, they are called Kupffer cells. So you need to remember this. It can be asked in some other way. So that's all for today. Before closing this particular class, if you have any doubts, please ask. And actually, I want to show you one more thing before we leave. So please ask your queries if you have. I'm trying to open one new window so that I can tell you something very interesting. So we have discussed about the hematopoietic stem cells and various kinds of cells which are produced from these, including neutrophils, eosinophil, basophil, red blood cells, platelets, monocytes, macrophages, dendritic cells, B cell, T cell, natural killer cells. All these cells are produced from the same cell, which is actually called hematopoietic stem cell. Now, the site of hematopoiesis can vary according to the age of an animal. For example, when we were embryo, our yolk sac and then liver was the major site of hematopoiesis, right? First of all, the yolk sac, and then it come to the liver. When we were 3 to 7 months old, right? So the spleen is actually the site of hematopoiesis. All the cell types in the blood are produced in spleen. So please remember the sequence: yolk sac, then liver, then spleen, and in the 7th month it comes to the marrow cavity. Generally, the erythrocytes are produced, right? Then at the time of birth, mostly the bone marrow is responsible for producing this, but not only bone marrow, the spleen and liver are also needed during birth or when a baby is born. Birth to maturity, number of active sites in the bone marrow is decreases but retain the ability of for hematopoiesis. So generally the bone marrow size will decrease, but actually the site in adults, for example, the bone marrow or the skull, ribs, sternum, vertebral column, pelvis. So all these bones which are having which are actually the long bones, which are having this bone marrow, these are the major sites in adults for producing the different types of cells in the blood. And if you see this particular graph, actually this one, it will clearly demonstrate that how this is happening. So in the fetus, this is in months, right? Then after this, the time is given in years. So keep in mind. So hematopoiesis, first of all, in the fetus, it is happening in the yolk sac, and you will see this graph is decreasing around about 3rd month. The yolk sac is not producing the cells at all. Then it come to the after 1.5 month, there is a start in the hematopoiesis in liver and spleen, right? And around 4 to 5 months, it is showing the maximum hematopoiesis. Then the function is uptaken by the bone marrow, which are actually the axial skeleton. And as you see, this bone marrow, distal long bones, it will come down and down around 30 years. This will go down. But the axial skeleton, as we have discussed, the skull, ribs, sternum, vertebral column, pelvis, proximal ends, or the femur, all these bones are responsible for, and their bone marrow is responsible for producing the different types of cells in the blood. It's called hematopoiesis, right? So bone marrow of the skull, ribs, sternum, vertebral column, pelvis, proximal ends, or the femur, all these are responsible for producing the hematopoietic stem cells in all of us because we are adults, right? So this can be asked, anywhere. This is very, very important. You need to remember the sequence: yolk sac, then liver, spleen. Generally 3 to 7 months, you need to remember it is spleen, then it comes to the bone marrow slowly, slowly. All right, so that's all for today. If you do not have any queries,